Good afternoon, dear students. In the last lecture, we discussed about what are the different methods by which a recombinant vector can be introduced into a recipient cell, which includes transformation, transfection, transduction, microinjection, electroporation, use of agrobacterium tumefaciens, and sometimes by using a particle, what we call as particle bombardment method. Now here in detail we are going to study how a recombinant vector is introduced into recipient cell by the process of transformation, okay, and how cells get transformed. So let us start. So how a recombinant DNA is introduced and how cells can be transformed. So let us study. So once your recombinant DNA, that is recombinant vector, is ready. What is a recombinant vector? Say for example, if the vector which you are using in your experiment is a plasmid vector, then the plasmid vector is cut with the help of restriction enzyme. Then the foreign gene to be cloned, say for example, the gene which you want to clone is a insulin gene. Then the required insulin mRNA or gene can be used, or you can use you can prepare the gene by using gene machine. Now the foreign gene, if you want to cleave it, it is also cleaved with restriction endonuclease. Then the foreign gene is introduced into the vector, and the vector so formed is called as a recombinant vector because the vector is carrying foreign gene fragment to be cloned inside it. Right. So after preparing the recombinant vector, it is allowed to enter inside a suitable host so that the foreign gene inserted inside the vector can be expressed. So expression of the foreign DNA is necessary, and for that purpose the recombinant vector with the foreign DNA is introduced into a cloning organism. Right. Now originally, and suppose we are going to study here how it is introduced by transformation, right? The procedure for transformation of bacterial cell by recombinant DNA or recombinant vector was first developed by Mendel and Higa in the year 1979. E. coli cells, suppose your cloning organism is E. coli cells possess restriction enzyme. Hence, whatever foreign DNA enters inside the cell will be degraded. To escape from the degradation, the exponentially growing cells which are present in log phase, okay, are treated with calcium chloride at low temperature. Okay. Now, why? See, whenever, say for example, if we are talking about transformation in E. coli, when E. coli cell accepts the foreign DNA, okay, or rather we can say E. coli cell takes up a foreign DNA only when the cell is competent. When is the cell competent? The cell is competent towards the end of logarithmic growth phase and beginning of stationary phase. Right. Now, only when the cell is competent, some 8 to 9 enzymes are produced which are required for uptake of foreign DNA and insertion of that foreign DNA into the host DNA. Okay. Now when the cells are not competent throughout their life cycle, so they won't accept any foreign DNA when they are not competent, right? And when the cells are not competent, and if the foreign DNA enters inside, the foreign DNA may get degraded because of restriction enzyme. Okay. So in order that the cell accept the foreign DNA, the cells have to be made competent. We have to make them competent. How? The cells are treated with calcium chloride in cold temperature. And when E. coli culture is treated with calcium chloride, cold calcium chloride, and then we add recombinant vector DNA inside the E. coli cell culture. Okay. First, E. coli cell culture is treated with cold calcium chloride, with calcium chloride at low temperature, and then we mix the recombinant DNA into the E. coli cell culture. Okay. Depending upon time, what will happen? Recombinant vector, that is the recombinant DNA, will enter inside the bacterial cell. What happens when we treat it with calcium chloride? The cell becomes permeable. When we treat E. coli cells with calcium chloride, they become permeable, the pore size widen, and hence the recombinant vector can easily enter inside the cell. When the recombinant vector enters inside E. coli cell, the process is called as transformation. Okay, but E. coli cells are poorly accessible to DNA molecules. But treatment with calcium chloride makes them permeable to DNA. So when they become permeable, then only the recombinant DNA can enter inside the cell. So how the treatment is given? Growing E. coli cell culture are isolated, or you can say growing E. coli cells are isolated. They are suspended in 50 millimolar cold calcium chloride at a concentration of 10 raised to 8 to 10 raised to 10 cells per ml. So we take 50 millimolar cold calcium chloride and cells are added to it at a concentration of 10 raised to 8 to 10 raised to 10 per ml. The cells are then incubated at 12 to 24 hours to increase the frequency of transformation, and then the recombinant vector is added to this E. coli cell culture. Okay. Now, efficient transformation, and what do we wait for? We wait that the recombinant DNA gets inserted inside the E. coli cells. Efficient and the cells undergo transformation. Once the E. coli cell accepts the recombinant vector, or once the vector enters inside the E. coli cell is said to be transformed. Process is called as transformation. Efficient transformation takes only few minutes, and then after you just take it for granted that transformation has taken place in E. coli cells and the cells are plated on a suitable medium for the selection of transformed clones. Okay. So you treat E. coli cells with cold calcium chloride, incubate, then mix it with recombinant vector, and then after a few minutes plate the E. coli cells. Plating of E. coli cell is done on suitable selective media, which will allow you to select transformed cells or transformed clones. The frequency of transformation is very low. Say 1 transformed cell per 10,000 plasmid molecules. This frequency of transformation can be further increased by using special E. coli strains like SK1590 or SK1592, etc. Some specific conditions are to be followed during transformation. The transformed cells are suitably diluted. First, the cell culture is suitably diluted, and then it is thinly plated. It is spread thinly, or we can say spread on a suitable medium, so that each cell gets well separated and produces a separate isolated colony. Okay. So the E. coli culture treated with calcium chloride mixed with recombinant DNA, then plated, but plating is done in a very thin manner. Okay, you have to separate the cell culture. So thin culture is spread thinly on the surface of a specific medium, so that each colony gets separated, each organism gets separated and produces a separate colony. The medium that is used for plating of transformed cell, is so designed that it will help you to identify only transformed cell, or the medium will support the growth of only transformed cells. Only the transformed cells will divide and produce colonies. Okay. Now let us look at this diagram. So you have E. coli culture in log phase. You centrifuge the cells, that means you separate the cells. You require 10 raised to 10 to 10 raised to 8 to 10 raised to 10 cells per ml. And then the cells are resuspended in calcium chloride solution 50 millimolar and kept in ice. Okay. And then they are incubated at 37 degrees, of course. Okay. Then you have to mix. Once you have chilled it on ice, then you have to mix with recombinant vector. What you have to add? The recombinant vector. This is the recombinant vector which is mixed with the chilled E. coli cell culture. And then usually heat shock treatment is given, and then after heat shock treatment you have to thinly plate it. Okay. So plating on LB medium. We have written here plus ampicillin. So ampicillin is an antibiotic which is added to the medium, which will help you in selection of only transformed cells, only wanted cells. Unwanted, non-transformed cells will not grow in the medium. So medium used for plating is so designed that it will support the growth of only transformed cells. Now, why do we need to do so? Why do we need a medium which will support the growth of only transformed cells? Okay. Now, after transformation, there are possibilities that 3 different types of cells are present in the cell culture. Okay, we have taken E. coli cell culture, we have mixed it with recombinant DNA, allow the transformation to take place, but still, products that are formed after transformation are maybe of three different types. Number one, the bacterial cell has undergone transformation. You can see here, this is a bacterial cell. This is the chromosomal DNA of bacterial cell. The cell has undergone transformation. It contains the plasmid with insert. It contains the plasmid with insert. Plasmid with insert means what? Recombinant plasmid. Plasmid with insert means recombinant plasmid. That means here recombinant plasmid, that means the plasmid is having foreign gene inserted inside it. So you can see here a plasmid and a foreign gene inserted inside it. So that means bacterial cells have undergone transformation with plasmid having foreign gene inserted inside it. That means transformation with recombinant plasmid. So here in the first cells, plasmid with insert is present. So these cells are desirable. Now in the second type, you can see the cell has undergone transformation, but it contains plain plasmid. Recombinant plasmid is not present. The cell has undergone transformation with plasmid which is without insert. Plasmid without insert means plasmid without foreign gene. Such cells are not desirable. We do not want the clones of these cells. The third type of cells are those which have not undergone transformation. Okay, the cell is such that it has not undergone transformation. So you can see there cell is present but plasmid is not present. So only genomic DNA is present, there is no plasmid. So even the third type of cell is not desirable. The second type of cell is a transformed cell but the plasmid is not having foreign gene. So we don't want these cells, clones of these cells. Okay, and then the third type of cells are non-transformed cell which do not contain the plasmid, so they are not desirable. So how do we plate the cells after transformation? When you want to identify the transformed cells, the cells are plated on the medium. And the medium is so designed that it will help you in the selection of only the first type of cells which contain plasmid with insert. Okay, it will help you in selection of transformed cells, cells transformed with plasmid having insert. And that is why the next step after transformation is identification of transformed cells. What is the next step? Identification of transformed cells. And in the next lecture, we will discuss about how cells that have undergone transformation or transformed cells are identified. Okay. So, identification of transformed cells we will discuss in the next lecture.